Hi guys, welcome to lecture 2. We are going to talk about the immune system. So there are two main types of defenses in the immune system. We have the non-specific, which is innate, and then we have the specific defense, which is related to our B and T cells. So a pathogen is a biological agent that's going to cause disease or illness in the host. So it can be bacteria, it could be a parasite, it could be a virus. And an antigen is any foreign substance that can cause an immune response. So whether it be a toxin like a bee sting, an allergen like a pollen, or a bacteria and any protein that they release, or a virus. And then we have lymphocytes, which are also called leukocytes, and these are basically any white blood cell. So let's talk about the nonspecific innate defense. So these defenses are not specialized for any particular invader. So the nonspecific defense refers to those that protect against any kind of pathogen. So plants, invertebrate, and vertebrate have nonspecific defenses. And these occur to all pathogens, and there is no memory involved. This is important. No memory involved. That's how we know that it is nonspecific. So for instance, our skin. These are nonspecific defenses. Right here, skin, our mucous membranes, chemicals, etc. They basically decide what comes in and out, and they deter other things. Okay? That's the first line of defense. A second line of defense is phagocytosis. Inflammation, fevers, etc. They occur due to exposure. And then our specific defense is our third line. These are going to be our lymphocytes and our white blood cells, and specifically in vertebrates, the B cells and T cells. So let's look at some examples of innate or nonspecific defense. First line of defense, again, we've got our barriers to pathogens. So we've got our skin, epidermis in plants. We've got our mucous membranes that are going to trap particles like in your nose and such. And then we also have our gastric juices in our stomach. Then we have chemicals. We have sweat. Plants have wax. And again, acid in stomach and lysozymes that are going to break down bacteria walls. Then we have symbiotic bacteria that is found in our digestive system. And basically what happens is they do us no harm, obviously, because it's symbiotic, but they outcompete the bacteria that invades our stomach that can cause illness. Then we have our second line of defense. And our second line of defense starts with our phagocytes. And basically they have the ability to engulf foreign particles. And there are large phagocytes that are called macrophages. So this is an example of a large macrophage. Then we have the complement system, and notice that it is not spelled like regular complement. It is a group of proteins that when activated, they basically form a membrane attack complex in a pathogenic cell. And this is found in most living things. So for instance here, this is the complement protein, and they are forming that membrane attack complex in that cell. Because this is actually showing, you can see here, it's the plasma membrane of an invading microbe. Then we have interferons. These are chemicals produced by a virus infected cell. And basically, those are going to signal to other cells that they need to produce the proteins to defend against the virus. Then we have a fever. And then we have the inflammatory response. And the inflammatory response has mast cells, and they release the histamine due to tissue damage. And histamines are going to cause the blood vessels to dilate and become the area around it to become leaky. And it's going to look red due to that increased blood flow. So this is, for instance, if you have even allergies, if you have a runny nose, things like that. But that increased blood flow causes an increase in temperature in the area. And then the plasma around the area leaks out, causing it to swell. And then that damaged tissue the chemicals released attract the phagocytes to engulf that foreign particle. And then the complement proteins leave blood to target cell membranes of bacteria. So here, this is showing that. Basically, here you can see we have a splinter, and it has breached the barrier, and it has pathogens all over it, right? So what's going to happen is that when this pathogen enters the area, then the mast cells, you can see they're releasing histamines. And then the histamines, you can see here, this is a regular blood cell, and after those are released, look how much bigger that blood cell got, right? So it's actually going to cause the blood vessels to dilate, and that area to start to look red, etc. And then you can see phagocytosis is happening here. They are killing all the cells out. So if we look at our specific defense, these are going to be defenses that utilize the B cell and T cell cells. And let me back this up here. The B cell is called humoral. The T cells are also referred to as cell-mediated. Okay, so let's look at the B cells. They are involved in the humoral response, and they work through the use of antibodies. And these B cells have a receptor that is called an antigen receptor, and they are antigen specific. So that means they can only bind to one antigen. So if an antigen binds, the B cell is going to divide or proliferate, and this is called antibodies. They can also be called or differentiated, into either plasma cells or something called memory cells. So plasma cells are going to produce antibodies. So the antibodies are going to bind to the protein or on the antigen and signal them for destruction. They're going to bind to the surface protein to inactivate the antigen, and they can also activate complement proteins. So let's take a look at these three things that the B cells can do. So first, we have the antibodies. So right here, here's a variety of B cells, and we've got this antigen receptor. So basically, when that antigen invades and it binds to that receptor, then it is going to come down here. Proliferation occurs, right? We're going to duplicate. So we've got more. And then the antibodies are going to be secreted into that circulation. So after it takes care of the cell, all of that, signal them for destruction. It's going to release those antibodies so then next time it will remember if that antigen shows up again. Then we have the surface proteins. Right here, this is a surface protein. And basically, it blocks the viral binding sites, right? So here's a virus, here's a bacteria. It's going to block it so it cannot bind. So it can't do what it came to originally do, which is bind and replicate. Then we have the complement proteins being activated over here. And you can see it's on a foreign cell, and it will cause the cell to lyse. Now let's look at this diagram here, and basically it is showing you over here on the left the antibody concentration over time. So we can see that the initial exposure here, it takes a little bit longer for a reaction to happen. Versus here, the second exposure to the antigen, the response is going to be faster and it's also larger. So that's why these cells are so important. So the memory cells of the B cells, they are long-living B cells. They do not release the antibodies, but they actually circulate and protect you from invasion quickly. So it's going to provide immunity to many diseases after that initial exposure. And as we talked about, that second response here is going to be much more rapid and enhanced. Then we have cell-mediated T cells. So the T cells actually target your cells. So here's a little diagram to help you remember. B cells attack outside the cell. T cells, they attack the invaders inside of your cell. And so, for instance, this is cancer cells, virus-infected cells, etc. So here we have a T cell. There is a virus-infected cell. It's going to clone those T cells. And there are multiple kinds of T cells. There are killer T cells, helper T cells, memory T cells, and suppressor T cells. I would not worry about knowing what each of those does. I mean, based on the name, you could probably figure it out, but just be familiar with the idea that there are multiple types of T cells. So our T cells can only bind to the proteins on the surface of an infected cell. And this binding site, which is a cluster of proteins, is called the MHC. So the MHC markers, which by the way, MHC stands for Major Histocompatibility Complex. These complexes of proteins on the surface are on every cell. And basically what they do is they allow your body to recognize your cells compared to foreign cells so that they do not attack your cells. So when a cell is cancerous or infected with a virus, it is a non-self proteins are displayed on the MHC complex. And this is going to trigger a cell-mediated or T cell response. So here we've got a T cell. This is, let me get here, this here is your MHC as well as the binding site. And here you can see in here, this is the antigen. And this is our virus-infected cell. You can see here, this is an MHC marker. And so this is a self-protein saying, hey, this is one of our cells. And then basically that MHC ends up killing that targeted cell. So there are three types of the T cells that we are going to discuss: the cytotoxic, the helper, and the memory. So a virus cell or a cancer cell is going to be destroyed by a cytotoxic T cell, also called a killer T cell. And this is going to release what is called perforin. So what's going to happen is the killer T cell or the cytotoxic T cell is going to bind to that infected cell. It's going to release that perforin molecule. And perforin does exactly what it sounds like it does. If you look at the base of the word, it's like perforated. It is going to form holes. And then the cell will lyse. Okay? So our T helper cells, they are going to bind to the antigen-presenting cells, and then the macrophages will ingest the microbes and destroy them, and display some of the foreign proteins on the surface. These T helper cells will also release chemicals that will activate the B cells to produce antigen-specific antibodies. And that's what these diagrams here are basically showing you. This is the helper cell. And then we have the T memory cells. And they are produced, they're long-living, they result in faster, stronger response. And you can see here what it's doing. It's got now a memory here of this virus in it, of the macrophage. Of the virus itself, so that if it happens again, it will be much faster. So we have two types of immunity that we are going to look at. The first one is active, and this is going to be your B and T cells. And then we also have what is referred to as passive immunity. So active immunity involves the body actively stimulating and producing those B cells and T cells in order to fight infection. We can stimulate the development of this active immunity, according to science, by giving vaccines. Vaccines contain inactivated viruses or bacteria, and that's going to lead to the production of memory B and T cells in order to prevent a secondary infection. Then we have passive immunity, which is actually lifelong. It's not lifelong. Active immunity is lifelong. However, it is not, vaccines are not lifelong. That's what I meant to say. So passive immunity involves transferring antibodies from one individual who's had something to another. So for instance, before birth, babies are protected by mom's antibodies that cross the placenta. After birth, breastfed babies actually get their mom's antibodies in the milk. Okay, then we need to talk about antibiotics. So basically, it's going to refer to antibiotics refers to the molecules that we get from bacteria or fungus that are harmful to other microorganisms. So for instance, penicillin, which is a type of fungus, produces a chemical that actually inhibits the growth of bacteria. So what they figured out is that they have this bacteria here, the staphylococci, and it's in a petri dish, and they noticed that whenever this penicillin, this mold was grown, that there was no bacteria growing near that. And that's how they figured out about penicillin. Okay, let's talk about viruses. We are going to focus on the life cycle of the bacteriophages that can lead to transduction. we're going to focus on the life cycle of the RNA, the retrovirus, that have high mutation rates, and then also that bacteriophages can respond to the environment. So let's look at the life cycle of a virus. So once a bacteriophage, which again, we talked about this before, but remember it's a virus that infects a bacteria. Once that bacteriophage infects a cell, two different life cycles or stages can occur. We are either going to have a lytic or a lysogenic. So the lytic cycle is basically, it's a lot shorter. It's going to lead to the production of the new bacteriophages that are released, and then it's going to lead to cell death. So basically, you can see here, here's our bacteria, here's our DNA. You can see the heads here are packaged with the DNA. They basically form everything that they need in order to reproduce and everything else, right? And then the cell lyses and it's going to release the infected bacteriophages. They are going to attach to a receptor site on another cell and then go through the process again and reproduce over and over and over and over again. Then we have the lysogenic cycle, and this is when the virus remains latent or dormant. And in this case, viral DNA is going to be inserted into the bacterial DNA, and then the bacteria cell divides, leading to many cells with that viral DNA. And then an environmental change or signal causes it to enter the lytic cycle. Retroviruses are going to contain RNA as the genetic information, not DNA. So an example virus, the most popular one that most people know about is HIV. These must also contain an enzyme called reverse transcriptase. And this reverse transcriptase is going to convert RNA into DNA. So viral DNA is then going to be inserted into the host cell. So the virus is going to utilize enzymes like RNA polymerase, as well as different organelles like the ribosomes, the RAF-ER, the Golgi of the host in order to create more viruses. So first, the viral DNA is transcribed by RNA polymerase to make messenger RNA. And there's two types of virus genome, and then the second is the messenger RNA that codes for viral proteins. And the viral proteins, they are going to include more reverse transcriptase, as well as capsid proteins, glycoproteins, etc. So you can see here, if we look, here's our host cell, and this is being released, right? So the retrovirus is released. Here's the reverse transcriptase. So what it's going to do, it is basically going to make a copy of our DNA, and then it will insert into the nucleus, it will be released, and it will go through the whole process, protein synthesis, everything that we do. It will go through that, and it will create more. So the HIV virus actually targets our T cells. And these are the ones that obviously help boost our immunity. So it limits the person's ability to fight off common infections. So HIV, as you know, turns into AIDS, and this occurs when the T cell count falls below a critical level. And this diagram actually shows you that. You can see here, this is the T cell count over here. When the T cell count gets to this low of an area, then there are infections, basic common infections, like viral, fungal, bacterial, parasitic, all of that this person cannot fight off, and it does end up leading to death. So this right here is AIDS. Now let's move on to bacteria. So bacteria has the ability to communicate and respond through a process that is called quorum sensing. So quorum sensing is when bacteria, through chemical signaling, can express different genes depending on the density of the bacteria population. So a detection in a high concentration of chemicals leads to activation or turning on of different genes than when the bacteria has a low population density. So you can see here, this is a low density versus a high density. And you can see that these genes, there are now new genes expressed compared to over here. So this is, when it's aggressive, this is harmless. So basically what they do is when we're in low concentration, the host is fine. See the smiley faces? We're fine. We're happy. It's okay. When this density increases, now the host is unhappy. And here, this is when they actually start to attack the host cell. So basically, they invade, they spread, they conjugate, etc. There is such a thing called quorum sensing and biofilm production. So a biofilm is a group of microorganisms in which cells stick to each other on a surface, and then they adhere to cells, and they are frequently embedded within a self-produced matrix of the proteins. So the cells in different areas are going to express the different genes, leading to cells with different functions. So basically what happens here is that they attach, they colonize, and as they're colonizing, they develop this biofilm. So this makes it very hard to treat them because of this matrix that's produced. So we have them, for instance, biofilms form on teeth called plaque, implants for hips, knees, etc., and even ulcers are formed from a collection of bacteria. So it's important that you realize that biofilms also form in nature and other things. And that, my friends, brings us to end of lecture 2.